this unit, we're going to learn about herpes viruses. And that's one word, herpes virus, and it refers to a family of viruses. There are a lot of different kinds, but not surprisingly, the ones we care about are the 8 that infect humans. And we call those the human herpes viruses 1 through 8, or HHV 1 through 8. So when people say herpes in common parlance, they're usually referring to herpes simplex virus 1 and 2, and those are actually just HHV 1 and 2. And as you can see, there are actually these 6 others that also infect humans. Learning about them can be kind of hard because they have so many names, but we're going to refer to them by their most common names here in the middle column. And these viruses are ubiquitous and not seasonal, and you can really get them anytime, any place. As an example, somewhere around 90% of adults have gotten CMV, 60% have HSV1, 20% have HSV2. Now let me give you the 411 on herpes viruses and their structure and replication. So starting with structure, they are all double-stranded DNA viruses, which means a low mutation rate. Now structurally, the DNA is in the viral core surrounded by capsid, and then around that there's tegument, and around that there is an envelope. And there are glycoproteins that are in the envelope and extend beyond it. And you might think that an envelope would protect a virus, but it actually doesn't. Envelope viruses are usually more labile or less stable in the environment. So herpes viruses will actually not hang around in the environment that much. Now here's one critical fact to understand about herpes viruses. So when a viral particle goes into a cell, it goes to the nucleus where it deposits its DNA. And when it does, it faces a choice, either to become lytic or to become latent. And lytic means that the virus is going to make new proteins, replicate its DNA, and then create new viral particles that can go infect new cells. And that's probably what you're used to thinking of a virus as doing. We call that lytic because all the progeny virus that's produced ends up killing or lysing the cell. Now latent means that the viral DNA silently settles in the nucleus and does not start replicating and transcribing genes. And it can actually stay there for years. And the question is, why would the virus want to do that? And the answer is, at some later point, it can reactivate. We don't know exactly how or why it reactivates, but something happens and the virus starts transcribing some of its genes again and gets the whole lytic machinery going.
Herpes, also known as HSV1 and 2, is a viral condition that affects millions of people around the world. Herpes symptoms can be painful, embarrassing, and irritating. The clearest symptom of herpes is the formation of lesions around the affected area. The primary herpes infection is often the worst, with subsequent outbreaks becoming lighter and less frequent over time. How is herpes spread? Herpes is spread through unprotected contact with someone currently shedding the virus. Generally, this occurs through kissing or unprotected sex. What is the difference between HSV1 and HSV2? HSV1 is the herpes virus that has adapted to affect the mouth. HSV2 is the herpes virus that has adapted to affect the genitals. When you have HSV1 of the genitals or HSV2 of the mouth, you're still contagious and can still spread the virus either to the mouth or genitals of someone else. Additional information on HSV2: 16.2% of the population between the ages of 14 and 49 have genital HSV2. 70% of those that get herpes got it from a partner when there was no sign of an outbreak. 2/3 of those infected with HSV2 do not know they're infected.
The herpes simplex virus has a large double-stranded DNA genome that carries the genetic information for the synthesis of over 100 polypeptides. A protein capsid surrounds the DNA, and a phospholipid membrane surrounds the capsid. The virus has elaborate glycoprotein spikes that allow it to recognize, bind to, and fuse with a few cell types in the body. The viral envelope becomes incorporated into the host's membrane, and the nucleocapsid uncoats and releases the DNA into the nucleus. In the nucleus, the DNA circularizes, and the host enzymes begin to transcribe the first sets of viral genes, known as early genes. The early mRNA molecules enter the cytoplasm, where they're translated into early proteins. The early proteins, in turn, enter the nucleus, where they're required for viral DNA replication. At this point, the cell begins to transcribe late mRNA molecules. These mRNAs enter the cytoplasm for translation into late proteins. The late proteins include the capsid structural proteins, which enter the nucleus. The late proteins also include envelope proteins, which are produced in the endoplasmic reticulum, ER, and become incorporated in the nuclear membrane. The viral DNA continues to replicate. One model postulates that the viral DNA replicates by a rolling circle mechanism. During this latter stage of replication, end-to-end copies or concatemers of the viral DNA form. These copies are cut into individual genomes as they're packaged into capsids. At the nuclear membrane, the capsid and DNA bud off and become surrounded by membrane material with embedded membrane proteins. The virus passes through the endoplasmic and buds off this membrane before migrating to the plasma membrane. The virus fuses with the plasma membrane and is released from the cell.